Prostate cancer is the most common cancer in Australian men, with 1 in 5 being diagnosed by the age of 85 and over 3,000 deaths annually. The cancer develops in the prostate gland due to the recruitment of reactive stroma, which release pro-tumorigenic factors at sites of epithelial inflammation, leading to uncontrolled proliferation and the development of the phenotype. An important hallmark of cancer progression involves the change of physiology from oxidative phosphorylation to aerobic glycolysis. Late-stage metastatic prostate cancers revert to aerobic glycolysis and produce large concentrations of lactic acid, which is then secreted into the surrounding tissue environment. This secretion has been hypothesized to play a key role in local tissue invasion and metastasis. The first-line approach for metastatic prostate cancer is androgen deprivation therapy. However, patients will often become highly resistant to a continuation of ADT. This type of cancer is called castrate-resistant prostate cancer, and we believe that our therapy could present a promising new method for treating this.
As previously mentioned, castrate-resistant prostate cancer cells exhibit reprogrammed cellular metabolism from oxidative phosphorylation to aerobic glycolysis. This switch, whilst of a lower energetic efficiency, uses significantly increased concentrations of glucose and in turn produces significantly increased concentrations of lactic acid. The overproduction of lactic acid is secreted from the cell through the monocarboxylate transporter 4 plasma protein, or MCT4. The work of our colleagues at the London Institute of Cancer Research has shown the MCT4 protein to be overexpressed in HER2-positive breast cancer models. The inhibition of the MCT4 protein has successfully reduced the proliferative ability of breast cancer cells. Through targeting the MCT4 protein and inhibiting the reprogramming of cellular metabolism in castrate-resistant prostate cancer cells, our team believes we have found a novel and effective therapeutic alternative for the treatment of a currently untreatable disease. We've designed an antisense oligonucleotide, or ASO, which binds the mRNA strand following transcription of the MCT4 gene. In normally functioning individuals, the DNA segment containing the MCT4 gene would be transcribed to produce a single-stranded mRNA sequence, which is then translated to produce a functioning MCT4 protein, which is translocated to the membrane and aids in the removal of the overproduced lactic acid from cancer cells. Our ASO will bind to the mRNA sequence, inhibiting translation and thus preventing the production of a functional MCT4 protein. In turn, there will be a buildup of lactic acid in the cell. By inhibiting the removal of lactic acid, we expect to inhibit the uncontrolled proliferation ability of the prostate cancer cells, as seen in the HER2-positive breast cancer model. By inhibiting the proliferation of the prostate cancer, we in turn expect to inhibit metastasis and neogenesis. The synthesis of ASO sequences is already a well-explored technique. We are proposing an intraperitoneal injection into the prostatic region containing sufficient concentrations of our ASO. During the pre-hybridization period, the ASO will be taken up into the cells via endocytosis. Following this, the ASO will bind to the target sequence mRNA strand in the nucleus. And finally, during the pre-hybridization period, there will be a degradation of the target mRNA sequence and inhibition of MCT4 production. We would also like to mention the use of different ASOs alters the outcome of the target mRNA sequence. We suggest future research into the usefulness of degradation, disabling, and modification of the mRNA sequence could produce more effective therapeutics.
We hope to reduce potential side effects associated with inhibition of cellular evacuation of lactic acid in the liver, heart, and kidney by delivering our treatment directly to the prostate. Targeting the prostate may result in infertility, as it may disrupt normal prostate function during ejaculation. Moreover, since the prostate is associated with the urinary tract and bladder, any treatment targeting the prostate has potential to cause irritation in the bladder and affect urination. As previously stated, the current method of ADT is insufficient. For this reason, our unique approach in targeting the metabolism of the cell provides a potentially successful treatment in combating prostate cancer. By disrupting the lactic acid secretion pathway by targeting the relevant MCT4 transporter, we hope to provide a novel therapeutic target by inhibiting the deregulation of cellular energetics. The use of ASOs to target MCT4 have been shown to ameliorate the cancer phenotype. This is evident in the pro-apoptotic and anti-proliferative characteristics, as well as decreased lactic acid secretion effects. The consequent buildup of intracellular lactic acid concentration is thought to be the driving force behind aerobic glycolysis inhibition. With the newly discovered anti-cancer properties of MCT4 ASOs and the known successful benefits of androgen deprivation therapy, investigations into the use of both treatments in conjunction could be the next step in finding a cure for prostate cancer.